Great. So it gives me great pleasure to introduce Sean Hoyan, who is the speaker this evening, and he's going to be talking about lichens. And Sean is the, actually, I don't know when you started being the curator of botany at the Nova Scotia Museum, but relatively recently, I imagine, where
I
at my
started
age,
in December 2018 is when I started.
Okay, I was going to say at my age, anything that's within 10 years is recent. Anyways, yes, so I think this is exciting. We have 57 people listening in, which is I think above our average. So I think there, as Sean and I were talking about a little earlier, there seems to be an increasing interest in the groups of organisms that we have tended not to pay so much attention to, at least as a society. So so it's going to be wonderful to hear about lichens this evening. So Sean, take it away.
Thank you, Soren. Good evening, everyone. Thanks for inviting me out to tell you a little bit about one of my favorite groups of tiny green things. As you mentioned, I started at the Nova Scotia Museum in 2018 as the curator, but I spent a little bit of time in Nova Scotia before that working as a postdoc at St. Mary's with Karen Harper, looking at cyanolichens, which are lichens that have a cyanobacterial photobiont partner, and how they relate to forestry. And before that, I looked at the relationship of bryophytes and other things growing on rotting wood in the forests to the type of thinning that's being done in some forests of northern New Brunswick. And before that, I was in British Columbia looking at lichens and mosses and caribou winter forage range. There's just they're a really interesting group to deal with ecologically and morphologically. For those of you who have a microscope at home, you probably have some sense of what I'm talking about. You get a little piece of moss under that microscope and you zoom in on it, and it's like you're looking at a whole different world, a little alien planet with critters walking around and all sorts of different crazy looking trees. It's like something out of the Dr. Seuss book. So that's kind of why I zero in on these groups as a taxonomic focus for myself and as a career. They're a really neat group to look at, and they're physiologically and evolutionarily really interesting too. Anyways, I'm going to get into it because I cover some of that in the talk. There will be time for questions at the end. So if you have an absolute burning question and you don't think you're going to be able to remember it at the end, feel free to interrupt me. But otherwise, do your best to save it until the end, and there should be time to talk about it. All right, I'm going to attempt to share my screen here. How's that working? Everybody see the title slide? It should be black and white.
I think it looks good, Sean, certainly on my computer.
Good. Okay. And I'm just going to test the advance. Is that changing for everybody?
Yep.
Yeah.
Okay.
It's working. Good.
Good. All right. Here we go. Lichens in Nova Scotia. So not everybody is in Nova Scotia is a biologist or someone who understands what lichens are biologically, and nor should you be. We value diversity in backgrounds as we value diversity biologically. So I'm gonna start in on this a little bit easy for those of you who want that very basic background on what are lichens biologically, ecologically, how do we classify them, how many species do we have in the region, in the province, what is it about Nova Scotia, why are we hearing about them all the time lately, a little bit about species at risk. Then I'm going to get more into my area of focus as a researcher, lichen conservation, what they're sensitive to, what things threaten them as a group. I'll talk a little bit about what is being done generally in Nova Scotia, but then I'll focus more on my own work during the presentation. I'll end with a few slides if there's time on how you can get into doing a little bit of lichen hunting yourself. So Again, if there are additional questions, please do your best to save them till the end. All right, what is a lichen? I like to think of it as a photosynthetic fungus. So taking a step back here, I wish there was an easy way to do a quick poll here, but I'll just ask you to respond in your mind. How many of you have heard of the theory of endosymbiosis? So this is the idea where certain components of our cells are the result of an evolutionary merger between two organisms. So when we think, a lot of people define lichens as a symbiosis, a partnership between a lichen and an alga, or a fungus and an alga, or a fungus and a cyanobacterium. But I think that that's kind of an odd way to look at an organism that doesn't exist in any other way in nature. So it's true that the algae and cyanobacteria can be free living, but the fungus never is in nature. It has to have the cyanobacteria or algal partner. So it is a species, it's a proper species. It doesn't look that way in any other combination. And yet we most often talk about them as symbiotic organisms. So I prefer to refer to them as photosynthetic fungi, because to me, they're kind of just doing, taking another approach to some sort of endosymbiosis, in the same way that the cells that make up photosynthetic plant cells, or the cells in our own bodies with mitochondria, ribosomes, the same way that they have merged with bacterial cells way back in evolutionary time to create what we have now. Lichens have discovered a successful strategy in merging a fungus and an alga, and they've been at it for hundreds of millions of years. So what are lichens? Ecologically, morphologically, they're what we call poikilohydric organisms, meaning their internal moisture content fluctuates with that of the environment. They're like a little sponge. If it's hot and dry, the lichens are going to dry out. Plants can't do this, or at least not vascular plants, the things that we have in our garden, the trees. When they dry out, they die. Lichens have a bit of an advantage there. They're, on the other hand, they are slow growing because they don't have roots and a large network from which to gather nutrients, and a large photosynthetic apparatus from which to sequester carbon. What you see is what you get. They have to get everything through that surface, so they're slow growing. They also tend to be very long-lived, that's part of the reason we have some conservation problems with them. Interestingly, they're reproductively flexible. I read a paper recently where the authors suggested there were as many as 27 different methods of reproduction in lichens. I can't name them all for you. I can name a few, but that's a lot of different ways of reproducing. They're chemically really diverse. So one of the ways that we identify many of these lichen species is by doing little spot tests. We'll take a drop of a basic chemical, often like common household bleach is one, potassium hydroxide is another. There are a few different ones we'll use, and it'll turn a different color when you put that spot on lichen of the right species. So that's like this chemical fingerprint is one of the ways we identify them. It's also useful for a number of reasons. People have used them as medicines, as clothing dyes, lots of interesting uses there. So in terms of classification, or how we talk about lichens, how we group them, well, we can group them by their type of what we call photobiont partner. So that's the thing that actually does the photosynthesis in the partnership, either the alga or the cyanobacteria. And there about 90% of lichens are what we call chlorolichens, which means they have an algal partner. About 10% are what we call cyanolichens, meaning they have a cyanobacterial partner. And these partners are nestled right within that fungal mesh of the lichen itself, and they just go about their photosynthesis, and the fungus soaks up all the sugars they produce. We can classify them by growth form. In general, the broadest growth forms we can talk about are basically crustose, foliose, and fruticose. Fruticose, you might think that's a bit of an odd term. Another way to say that is just shrubby. It's I think it's an old Greek word that just means shrubby. So foliose, that makes more sense. Foliage, you're used to hearing about that. So that's leafy, and crustose, obviously crust-like. So we have crust-like, leafy, and shrubby lichens. And you can see examples in the pictures at the bottom of the screen there of what those different growth forms look like. We also group them by substrates. Substrate meaning what they're growing directly on top of. We can call them epiphytic. And these on either side of these slash marks in this set of terms here, it's basically the Latin and the Greek version of the ways that we refer to them. I don't know why we use both Latin and Greek to refer to the substrate associations of lichens. Presumably some scientists wanted to be fancy and some competing scientists wanted to be fancier and they got into it, and I don't know how these things work, but anyways, there you have it. Epiphytic corticollis basically means growing on trees. Epizyllic or xylocolous, you see that xyla in both terms, basically means growing on xylem or wood. Epilithic or saxicolous basically means growing on rock, and epigeic or terricolous basically means growing on the ground. So we can group them by what they're growing on. There's a very small number of lichen species that are aquatic. We have one in Nova Scotia, a couple more that are kind of semi-aquatic, but that's debatable. Mostly lichens are not aquatic. Lichen diversity, how many species? Well, globally, we're not entirely sure because we don't know them all yet. As I'm sure you've heard with many other types of organisms, we're still counting, we're still describing. We think last I checked, that this was a couple of years ago, I put this number together, though, so there's probably more now. There's about 17,000 known at the time. In Atlantic Canada, we have about 900 known species, probably closer to 940 now. The main genera, or plural of genus, the main genera that we know of in Atlantic Canada, the most species-rich genus is Cladonia. These are the little white colored lichens that you see growing on the ground, on rotting wood, on the bases of trees. They look kind of shrubby. British soldier lichen is a good one. It has those little red dots at the top. 73 species of those guys in Atlantic Canada. Usnea is the next most species-rich group, 25 species. And probably a few more in there that we haven't found yet in the region for any of these genera, actually. Peltidra, following up on that. So the ones that are highlighted in blue here are cyanolichens. So as you might not expect this pattern that there would be so many species in these cyanolichen genera based on that 90%, 10% split of algal lichens to cyanolichens. But I think it's just that the classification of a lot of these things is still kind of expanding and changing a little bit, and we haven't, for the time being, those seem to be the patterns, but it's definitely changing a lot in the last few years and will continue to change. And so it's unlikely we will have such dense concentrations of single genera, of species in single genera from 10 years from now, for example. Then there's this division that we call macrolichens and microlichens. So basically, macrolichens is anything you can identify with a hand lens and a microscope, like a dissecting microscope, and maybe some spot tests. There are some exceptions to that. Usnea, for example, often needs thin layer chromatography, which is a specialized chemical test to distinguish different species. But in general, these macrolichens, these things that we see that they're large enough to see most of the morphology with the naked eye or a hand lens. There's about 341 species in 85 genera. That was, so that's Nova Scotia as of 2014. I think it's closer to 365 now, somewhere around there. Microlichens would be everything else within that 900 species known from Atlantic Canada. So microlichens would be the crust lichens, the caliceoid lichens, the very things that you need to look at the spores basically to identify in most cases, or do some sort of squash mount with a high power microscope. Lichen diversity. So as Nova Scotia special, you guys have probably been hearing a lot about lichens in the news lately, conflicts with logging, and just in general. Well, much of Nova Scotia is what we consider to be cool temperate rainforests. You probably don't normally think of Nova Scotia as being in a rainforest, but it is. Climatologically, much of Nova Scotia is considered rainforest. I can't remember what the exact cutoff is, but it ranges from, I think, it's a fuzzy cutoff somewhere between 1,100 millimeters and 1,400 millimeters of precipitation. Somewhere in that range is the cutoff for where you're considered to be a rainforest versus sub-rainforest climate. But cool temperate, some people also call it pure humid hemiboreal rainforest if you want to get super fancy. Basically mean annual temperatures 4 to 7 degrees with a low daily fluctuation, although you wouldn't know it from the last 6 weeks or so we've had here. Annual precipitation, there you have it, more than or equal to about 1,250 millimeters, often much more. We get up to about 1,900 millimeters of precipitation in some parts of Nova Scotia. and no dry season. There's kind of a really important feature, which means minimal drought type of disturbance, minimal fire disturbance, all the things that go along with dry seasons. Which means we have habitats that have a long term continuity, a lot of old growth forests when we let them be. And I think the record now is up to 520 years. I'm sure a lot of you folks have heard about our new record, longest lived tree, an eastern hemlock from the Pinook Lake Nature Reserve, or very close to it, was found last year. So there you have it. The dark gray regions in the map show the distribution of cool temperate rainforest in eastern Canada. Now, I think that sort of finger-shaped projection sticking inwards into northern Quebec there, I think that's a little bit of climate anomaly because of the lack of weather stations in that part of the world. But the rest of the region is, I think, a pretty accurate reflection of where the cool temperate rainforest belts are. So back to this continuity idea. Continuity is really important when we're talking about biodiversity. It gives more time for things to arrive in those habitats, and those habitats end up becoming very structurally complex. You have tall trees, you have short trees, because tall trees fall sometimes, create gaps, you get short trees. You get tip-up mounds, these big root plates that are vertical soil things. You get little wet depressions forming on the other side of those tip-up mounds. So you get this really high habitat heterogeneity, and you end up with a lot of diversity, both because of this time and this habitat heterogeneity. Also, as I mentioned, because lichens are slow to mature, sometimes as you can imagine, if you're relying on little things floating through the air, falling during rainstorms, those things have to germinate. If it takes 10, 20 years to reach maturity, then obviously you need, and you can't colonize a habitat until it is like an old growth forest. You know, some of these lichens are going to need forests to be at least 100 years old before they even get a foothold in. And some of them even need other species to prepare a surface that are also old growth associates. A good example of that is boreal felt lichen, which is an endangered species in Nova Scotia. We think that it is reliant on having these other things living on the trees, mix of liverworts and lichens that use some of the same cyanobacteria as they do before the spores can land on that tree and actually grow into it, resynthesize a new lichen. So here are photos showing Frulania liverwort and salted shell lichen, also known as Cococarpia palmicola. And that's on the left, and then boreal felt lichen on the right, the green thing with the little pink dots. That's boreal felt lichen. I mentioned the microhabitat diversity. Here's some data I collected when I was doing surveys for white-rimmed shingle lichen in New Brunswick. And they really like these leaning cedar trees in New Brunswick, where and they typically colonize the upper surface, this white-rimmed shingle lichen, which is a threatened species in Atlantic Canada. You can see that in the figure beside the photo of the tree, it shows the mean lean angle of the trees that we found this lichen on from New Brunswick and Nova Scotia. So clearly likes a tree with a good lean to it. And why? Well, my current hypothesis remains yet to be tested, but it's that it receives more liquid water on the upper surface of that trunk. And there is evidence that many of these cyanolichen species do require liquid water inputs. They can't just have humid air. It could be 90% humidity. These lichens don't care. They need that liquid precipitation to actually to become photosynthetically active. There's another very specialized microhabitat. This is very difficult to see. I'm going to move my cursor and hopefully, can you guys see that cursor moving?
Yeah.
Okay, good. So you can see there's some little tiny white pinpoint pricks here. This is a rotten into little rot hole inside an old maple snag, in which we find this stubble lichen called frosted glass whiskers, which is a species of special concern in Nova Scotia. It only grows in rotten holes in old maple trees. So some very specialized habitats for these rare species. Now, global context, Atlantic Canada is actually doing, compared to some places in the world, not too bad. We do have our conflicts, we have our problems, we have things we need to make better, but relative to parts of Europe, some of the New England states south of us, we're not doing too badly. And that has to do with the intensity of pollution for the most part in these lands, as well as the longer history of active management of forests. So in much of Northern Europe, you just don't have a lot of natural forests that haven't been logged at some point in the past, or that haven't been allowed to obtain these old ages. Again, we're not perfect here. We certainly we have a lot of room to grow, but relative to much of Europe and the New England states, where they've had much worse acid pollution, acid rain, and a much longer and more expansive history of disturbance and regular disturbance, if we're not doing as badly. But we do have some species at risk in Atlantic Canada, and here are the most recent ones. So the ones that are highlighted in red are the two endangered species. That's vole ears lichen and boreal felt lichen. The ones that are in orange are threatened species. That's black foam lichen, eastern water fan, which is the one aquatic lichen that we have in Atlantic Canada, and wrinkled shingle lichen. Some people call it wrinkled shield lichen. And then just in white, but bolded in orange, is white rimmed shingle lichen, which I think is it's been listed as threatened by COSEWIC, or recommended as threatened by COSEWIC, but it hasn't officially been added to Schedule 1 of the Species at Risk Act. I've heard through the grapevine that this is pending and will be very soon added. And as far as I know, most of the jurisdictions are treating it as this is going to happen. So that will soon be a full orange one there. Then in yellow are the two species of special concern, federally listed species of special concern, frosted glass whiskers and blue felt lichen. Ghost antlers antler lichen was assessed for New Brunswick and Nova Scotia and Quebec and found to be not at risk. So we do sometimes find that after we have a better look at the data and do some additional surveys, that these things are not as rare or uncommon as we thought they were, and are not necessarily deserving of the special status federally. So there's definitely a pattern here, though. You'll notice those blue arrows on the right side of the screen. Those are all cyanolichens. Remember that 90%, 10% split we talked about before? So this is definitely a disproportionately cyanolichen-based list, this list of species at risk. There's something about cyanolichens that makes them more susceptible to human activity on the landscape. So what are some of the threats that affect these lichens? Well, logging and conversion of old growth forests into other types of habitat, and logging of forested wetlands in particular, those are some pretty significant threats. There are more protections for them now. Many of you have probably heard about the recent conflict close to where you guys are based, where people found some rare lichens in this old growth forest that was set to be logged. And that's resulted in a number of patches within that forest being protected up to a 100-meter radius. Obviously, when something is being logged, if you cut down the host tree, the lichen's not going to make it. The lichens grow on those host trees for a reason. It's because they can't grow on the ground. They sometimes can grow on rocks, but usually they need that particular type of tree to grow on. So if you damage or remove that tree, they're not going to make it. There's also these indirect effects of logging. You can change the microclimate of the site, even if you're only doing partial thinning. Sometimes the habitat just gets too fragmented and they can't any longer disperse to different trees because the nearest trees that are suitable are too far away. There may also be interactions with other threats, secondary threats, which I'll get into in a moment here. And the recovery time may just not be enough. So if you're running forests on a 40-year rotation and the lichen requires an 80-year-old forest before it can even colonize, and then it takes 20 years to reach maturity, obviously those rotation times have to be longer. Some other threats, acid rain, I mentioned before, is one of the reasons that Europe and the northeastern United States have been hit so hard relative to what we've experienced here for their lichen impoverishment. Cyanolichens are more susceptible to acid rain than other types of lichens. People have done experiments that show this quite clearly. Invasive species, this is one of those secondary threats that interacts with forestry. So I'll talk about this a little bit when I talk about my research later on, but we think that these gastropods are having a big negative impact on lichens. You can see this picture of a tree here. all of the kind of olive brown color, that's a lichen. I think that's a lichen called Paneria conoplea. And all of the black part of the tree, that's where the lichen's been eaten away. And you can see the culprits right there. Little beige colored slugs over top of the lichen. Also, if you're paying attention to forest news these days, we've got a couple of big invasive pests coming our way or already here. Hemlock woolly adelgid, I think we're already in triage mode for that one. Obviously a big problem for old growth hemlock forests, and emerald ash borer, potentially a big problem for ash forests as well. We may also be experiencing reduced dispersal vectors. You may be wondering, how do these lichens get around? I mentioned they have 27 different ways of reproducing. Some of these ways that they reproduce are ejecting tiny little spores that can become airborne. And most of these spores are light enough to go aloft on wind currents and disperse quite widely. But some types of lichens will disperse by fragmentation or by larger asexual propagules that sort of pepper the upper surface of the lichen. And we think that a lot of these things are moved around by, well, we know that some of them are moved by slugs, by endozoochory, meaning they eat the propagules and then poop them out somewhere else on the tree, and the lichens can adhere and resynthesize there. In other cases, we think that they're accidentally dispersed by squirrels, birds, things that are up and down the trunks on a regular basis. And we know that certain types of birds, particularly aerial insectivores, are declining significantly right now. And many of these things live in the same habitats as some of the rarest lichens that we have, forests or wetlands in particular. So climate change, obviously top of mind for a lot of folks as well. And this is another threat to a lot of these lichens. So here are some climate change projections. And the blue and red one on the right is one that I made. The green, yellow, orange, and red one on the left, that's one that I think Troy McMullen did for the ecosioc status assessment of Paneria lurida. And you can see we do expect some range shifts for these lichens, and in some cases, so the one on the right is for blue felt lichen in Atlantic Canada. And in some cases, we do expect habitat to improve in the near future. So for blue felt lichen, we found that the greatest suitability throughout Nova Scotia is somewhere around the 2050s in the sort of more moderate climate change scenario, and going to be within the next 10 years in the more aggressive change for climate change scenarios. But thereafter, it seems to decline, depending on with rates changing depending on which climate projection you use. So with all of this information on threats we have on lichens in Nova Scotia, what do we need for research? Well, monitoring and inventory is kind of a given. We have to be out there watching these lichens and surveying understudied habitats to find out what other species we might have. We can't protect something if we don't know it's here yet. We want to know more about how detectable these things are by the people who are going out doing the surveys. Do we need to apply correction factors for the amount of ground that we cover, those sorts of things? We need to develop predictive tools. So biogeoclimatic habitat suitability models, like those maps I just showed you, those are based on relationships between the presence of the lichen and climatic variables, forest cover variables, things that we know are related to their ability to use a habitat. Habitat supply models, looking at how much logging we intend to do over the next several decades, how that relates to the distribution of key habitats in the landscape, and population size and trajectory models, looking at how we expect both of these other things to change and how that will affect the population sizes of the lichen. So there's a lot of mathematical ecology that needs to be done here. And also some more field-based ecology. We need to understand more about what are the vulnerabilities in these lichens recolonizing the trees that we know that they need. So are they dispersal limited? Are their vectors declining? In some cases, we think the juveniles or newly formed lichens, newly germinated lichens, are really susceptible to gastropods or slugs, snails grazing, because they haven't had a chance to build up that storage of anti-grazing chemicals in their bodies yet. And we need to understand more in general just about how threats and different things that cause mortality are affecting them on the landscape. And then applied conservation as well. So can we do transplants with these things? Can we Some of the things we've talked about in the conservation community are applying little exclosures to the bases of trees so the slugs can't climb up them. Some of this work has already started. We've tried doing some fertilization, we've tried doing some exposure experiments, and there's some interesting stuff coming up that I think is going to make a bit of a splash in the conservation world here in the next 5 to 10 years, and I'm excited about it personally. But these are the ones I'm going to focus on for talking a little bit more, going into more detail in this presentation here is more about monitoring and inventory, lichen detectability, and what specifically is threatening these lichens for different species and different scenarios so that we can mitigate those threats. So the first study I'm going to give you an overview on is, I call it my LACER study. And this stands for Lichens, Acidification, and Cut Edge Response, which is a tongue twister of a title that I liked it because Acer is the genus of maple trees in Nova Scotia, as I'm sure many of you know. And many of the lichens that are rare and at risk in our region grow on these red maple trees in forested wetlands. Cut edge response, so looking at the impacts of these clear-cut edges that are in our region, and acidification is an important feature, we think, affecting these habitats. So in this study, I'm taking a three-pronged approach. And I should mention this is in partnership with Karen Harper at St. Mary's University, and we've had a number of excellent undergraduate students and a postdoc, Hugo Maduros, who's also contributed a lot to this work. And we've collaborated with other groups in the region, like the Mercy Tobiatic Research Institute and the Atlantic Canada Conservation Data Center, with data sharing and exchanging as much information and helping each other out as much as we can. So the three-pronged approach is doing natural experiments, looking at the impact of existing clear cuts. We run a survey line from the edge of a clear cut into the interior of a forested wetland adjacent to it. And just see what the changes are with that line over time. We do a bit of modeling, looking at these impacts of human disturbance over large areas versus the impacts at the forest stand scale, and manipulative experiments. So we actually hauled bags and bags and bags of pelletized lime out to a couple of our forested wetlands and set them around the trees that we're monitoring lichens on. It was a lot of work, let me tell you. Those things are like, I don't know, 15 or 18 kilos a piece or something. The students who helped me out with that work were totally worn out at the end of the weekend. Anyways, our research questions: how important is each of these threats? So clear-cut edge, proximity, the presence of acidification or hyperacidification. At what scale do these threats operate? Are we talking about just needing to mitigate things at the forest stand scale, or do we need to think bigger? Do we need to think within a 1 kilometer zone of where those forested wetlands are, that we need to be balancing something in the landscape? Do the threats interact with each other? So thinking about logging and slugs and exotic slugs getting into those habitats. And we know that humans can facilitate movement of exotic invasive species. And can we fix them? So looking at some of that applied conservation stuff. So we started out with 16 or with 10 study sites. That's been
they put me into safe driving mode. So I'm trying to get it on Bluetooth. So I find, but that's it on there. What's happening?
Well, safe. Nothing wrong with that.
Is that a question?
No, but the computer decided this.
Oh. All right.
I can't hear it.
Simpsons, could you mute?
I should be able to hear it. I've got it here, but I can't. But I can't hardly hear him when he talks.
Do I need to speak louder?
People.
Yeah. I
I'll
can hear something.
hold my microphone next to my mouth.
Understand you.
Is that better?
I'm not. It's very sort of a bookish sort of a thing. But I thought I tried.
There we go. We good to keep going? Little technical difficulty there.
Carry on.
Okay. All right. So for this LACER study, we expanded our study sites throughout Guysborough and Richmond counties. I don't have those dots on the map yet, but they are there. And what we've so this is what forested wetland. This is one type of forested wetland. Anyways, there's lots of different types of forested wetlands, but basically we were focused on mixed wood swamps with 2 to 5 year old clear cuts on one side, preferably a southern exposure. Here's a little schematic kind of showing how we did it. We have these long plots that we had in kind of a nice little ladder shape for going from the clear cut edge all the way to the forest interior or the forested wetland interior at 150 meters in. A lot of these studies of edge influence in the past have only gone like 50 meters into the forest, and we know that's not enough. We know that people who have looked at it at deeper edge influences, whether it's from clear cuts or other things, they've found them. So we wanted to go as far as we could, and this is just based on the size of forested wetlands we could get for this work. That was about the farthest we could get. So we had seven survey plots in each stand and surveyed 3 red maple trees for lichens in each one. We looked at the amount of lichen cover and the diversity of lichens, a few bryophytes, and the amount of how healthy the lichens were and how much grazing there was on them. Measured a bunch of other environmental variables, including bark pH, and canopy cover of the trees. Basically, what's the importance of clear-cut edges relative to other things going on in that habitat is what we were asking. And for the most part, so this is a really complex diagram. Takeaway is look at those red lines and look at the black written labels at the end of those red lines. So if you look in the, I guess it would be the southernmost line in that little inset plot in that larger plot, distance from clear cut. It's not a small line, so that line indicates sort of the strength of the correlation with this compositional gradient in species we have. But you can see that it's less strong than the relationship with canopy openness within the plot and the relationship with the size of the tree bowl we were looking at. So there's a lot of things going on in these habitats that need to be accounted for before we can start to detect an influence from the edge of the clear cut, basically. And that doesn't mean that the edge of the clear cut is not an important influence, but it does mean that it's not the biggest influence on the composition of species in these habitats. And so we need, if we're trying to understand how that impacts lichens, we need to account for all these other things as well. But it is something we can detect. The importance is low for the community as a whole, but it is, these communities are dominated by generalist species like Loberia pulmonaria, tree lungwort lichen is the most abundant thing on these trees. And we know that one actually does pretty well in open forests. And so when that's dominating the community, of course, canopy openness is going to be the most important feature, not distance from the clear cut. But when we look at certain species, especially on the south sides of trees, we know that several species or groups are absent or reduced at the edge of these clear cuts. So here's that species I mentioned before, Paneria conoplea, the orange line. You can see all the way up to 80 meters until you hit 100 meters. You don't really have a spike in the cover of this species. And when you get to 100 meters from that clear cut edge, shoom, it's all over these trees. We lumped all the species of Usnea together because there's a lot of them that you can't tell apart in the field. And so we just lumped them together. But even with that as a genus group, you see that huge spike at about 80 meters from the clear cut edge. Now this is pretty early on in post clear cut development, so we can't necessarily attribute all of these changes to the clear cut itself. It could be to some extent a pre-existing gradient. We hope to clarify that as we do additional surveys as the years go by on these studies. And we do have some species that react positively to the proximity to the clear cut edge. Nothing does well right at the edge, right at 0 meters, because it's just been baked by the sun after being in an intact forest, and there's sort of a shock thing happening there. But species that are close to that edge, there are some species that like having perhaps elevated light and perhaps elevated proximity to other features in the habitat. Kalima subflachidium is one of the examples I provide here, the blue line on the graph on the left. We didn't really see any change of the health of the lichens. Bark pH didn't really seem to change with distance from these clear-cut edges, so at least clear cuts are not necessarily interacting with habitat acidification as we might have expected. But grazing damage, you can see this graph on the left, the blue line, grazing, the amount of grazing damage or grazing cover on these cyanolichens declines a lot as you get farther into the forest. There does seem to be a weird little spike at 150 meters. It jumps back up a little bit, but it's still, even there, it's significantly less grazing damage than when you're close to the clear-cut edge. So we think there is some interaction between logging and the grazing of these invasive exotic slugs. And we've done a little bit of trapping to look at how these slugs affect the lichens. We put together these tree traps, put them on all of our survey trees throughout the Southwest. We've added some ground traps as well. We got a lot more than slugs, all sorts of invertebrates, and we're working with project partners right now to get those identified. The long and the short of it is, it's mostly, when we're looking at just slugs, it's mostly exotic slugs. There are a very small number of native slugs that we were able to detect, but very few of them, or none of them, are exclusive to the trees, I should say. So there are none of these exotic ones that seem to have such a large appetite that are just staying in the trees. They're all going up and down because we catch them way more frequently on the ground than we do in the trees. So we know they're starting on the ground, going up into the trees, which sort of adds fuel to the idea that if we can do some sort of applied exclosure conservation for some of these really rare lichens, it might prevent them from getting overgrazed by slugs. There's that result again from our slug trapping experiment. So we see the grazing is higher at the edge, the slugs are more abundant at the edge. It seems like a pretty clear relationship right now. We also see an interesting pattern with wasps. I'm not sure what's going on here. I don't think it has anything to do with lichens at all. It's just an interesting pattern we saw from our trapping. Next steps for the LACER study is we're working on additional parts of this edge influence experiment. We were hoping to do gut content analysis of our trapped slugs, but that kind of, we don't think we can. They just aren't preserved in good enough condition by the time we get them to look at that sort of stuff. We have analyzed patterns of herbaceous plants as you get from the clear cut to the forest interior. That's another talk. And we've got our 6 to 8 new study sites identified in boreal-felt lichen habitat in Richmond and Guysborough County. So we're waiting to analyze those data and include them in the broader data set. We do also want to add some reference sites in undisturbed upland to lowland transitions. That's one thing I'm hoping to do more of this summer to look at, so we can really piece out what is clear cut, what is pre-existing ecological gradient in that change, and follow-up surveys down the road. This is me hauling large bags of lime out to start that liming experiment we started. And then we also have this cumulative effects analysis, the landscape scale that we're working on. We keep getting started on it, and then the person who's working on it ends up getting a job somewhere else. So we've got another postdoc who's waiting in the wings. She'll be starting in July. We're hoping she can get to us fairly soon. And we have started some interesting exposure experiments up in Guysborough County. So there's a lot of people involved in that project. Nova Scotia Habitat Conservation Fund, My Tax Elevate program, Port Hawkesbury paper helps us out with funding, NSERC, St. Mary's University, and the Nova Scotia Museum. Lots of people involved too. We're helping a lot of young biologists cut their teeth on field work with this project. I'm going to talk a little bit about canopy lichens. How am I doing for time, Soren?
It's 8:25, so you can take 10 more minutes or something like that.
Okay,
Be fine.
So this past summer, I got together. I was able to work with a wonderful undergraduate student who's very keen, and we got some funding again from the Nova Scotia Habitat Conservation Fund to look at canopy lichens. So you're probably thinking, well, aren't they the same as what's growing lower down? And well, the answer is we don't know. Nobody's ever looked at them really in Nova Scotia. I mean, people have looked at trees that have fallen over, maybe hypothesized, there's stuff higher up and that we're not seeing lower down, but we really don't have a good sense of it yet. People have done this in other places in the world, in British Columbia, Washington, State, Oregon State. People have done a fair bit of canopy lichen research, particularly in old growth forests, where you get these large old Douglas fir trees, and then even in the sequoias, the redwoods of coastal California. And you get some pretty interesting lichen communities up in the canopies. We also figured, we might not have sequoia out here in Nova Scotia, but we have some pretty long-lived trees. Eastern hemlock, which is, as far as we know now, the longest lived tree in Nova Scotia, can easily get upwards of 200, 300, 400 years old. We now know over 500 years old, even in some rare cases. It's at the northern edge of its range here, so perhaps not as big or impressive as you would get down in parts of New England or down in the Appalachian Mountains, but it is increasingly threatened by hemlock woolly adelgid, a little defoliating insect. And so we were kind of wondering, looking at the research that's been done elsewhere and the threats, these imminent threats or urgent threats to Nova Scotian old growth trees, are we looking at, are the species growing on these trees exceptionally vulnerable right now because of these threats to the forest sector? And are we, when we do our surveys all within reach of the ground for species at risk or biodiversity generally, are we biasing our population and diversity estimates low? In some cases. So these are important questions for conservation. So we decided to put together this study to look at it. We decided to use something called single rope ascension technique, where you shoot a rope up using a giant slingshot. Some people use a crossbow, some people use a rope gun. There's lots of different ways to do it. We use a slingshot, shoot it up over a branch high up in this tree, and you climb up to the lower crown on this little rope, wearing a harness. And we looked at the lichens. We compared them to the ones we found at the ground. And we did this in 3 general areas: the Southwest, the Central region, and Cape Breton in Nova Scotia. We wanted to get up into the Cape Chignecto area as well, but we didn't have time. So we surveyed 27 trees. Doesn't sound like a lot, but it takes a couple hours per tree when you're climbing and rigging and doing all the sampling and everything. We measured lichen cover and lichen diversity, some habitat features of the trees. And we found that indeed, we have more lichen species and in the lower crown of the tree, and those lichens are more abundant in the lower crown of the tree, particularly macrolichens. When we look at, we also looked at mosses and liverworts, so bryophytes generally, and we found they're richest at the base, as one might expect. There is this sort of general diversity gradient as well, from the southwestern Nova Scotia, where we have the highest diversity, up to Cape Breton, the more northern part of the range, where you have the lowest diversity. Interestingly, the lower crown of these trees has a higher pH, so it's more neutral, whereas the base of the trees tend to be a little bit more acidic. I mentioned before that a lot of lichens seem to be really sensitive to acid rain, so this raises, so we've confirmed that this pattern does exist in some trees in Nova Scotia, but raised the additional question: can the lower crown of a tree serve as a refuge for species that are sensitive to acid pollution? Should we be looking there more often for these rare species that have been knocked back in recent years by, we think, acid pollution? We're hoping to answer those questions in the next few summers. Katrina will be working for me again on this project this summer, and then she's starting a master's degree expanding on this project over the next 2 years at either St. Mary's or Memorial University. She hasn't quite decided yet on that. I mentioned pH, bark pH could be one of the reasons we see this difference here. There's obviously differences in light and moisture between the lower crown and the base of the trees as well. These are really old forests we sampled in there, 200, 300, 400-year-old forests. So there's been a long time for these gradients to get set in the forest more generally. We're also wondering, if we start looking at more hardwood species, particularly the things that normally host species at risk, are we going to find more species at risk in the canopy of the trees than in lower down? As you can imagine, as you get higher up in the parts of the tree you're looking at are younger, which means less time for colonizing. But if they're higher pH, maybe the habitat's more suitable. So there's some interacting factors here we want to understand a little bit better. And we also want to understand, we can't just expect everybody to start climbing trees to look for lichens when we're doing these biodiversity surveys. It's a really time-intensive process that it takes time. It takes a lot of specialized equipment and training. So is there, we want to look at how many trees do we need to sample from within reach of the ground to get the same number and type of species as we would get from climbing a few trees. Is there a trade-off there? Are there some things we only ever get in the canopy? We need to know a little bit more about that dynamic there. And can we develop some correction factors to help us more accurately estimate the number of species at risk that are in our province? And again, lots of people have helped us out with this project. Nova Scotia Habitat Conservation Fund, St. Mary's, NSERC, and the Nova Scotia Museum. Perpetual partners in lichen work. And this photo shows Katrina, the main student on the project, just getting up into the tree there, Miranda Fries, and our ground, we call it, arborists would call them groundies. She was our groundie this summer and helped us to stay safe and do equipment checks and all that. And I'll leave it there for questions. I do have some slides that I can get into if people want to know a little bit more about how to get into lichen observation, but you're also welcome to just reach out to me at the Nova Scotia Museum, where I'm happy to take on volunteers and anybody else who wants to learn more about lichens.
Excellent. Thank you so much for this amazing, informative presentation. I wonder if you say you might have some more slides, but I wonder if you could stop sharing so that we could get into the view where we can see everybody. I forget what
Absolutely.
it's called, but I think that might be a little easier. And this last part you talked about the lichens in the crowns of trees. You obviously must have read Meg Lohman's Arbonaut, her book on.
I have to confess I have not yet.
Oh,
read that
okay.
book.
Because
I recently heard about it, but I've not yet read the book.
Okay, well, you are. I think she's the forerunner when it comes to discovering the amazing biodiversity of lots of things in way up in the crowns, where she argues that most foresters, all they study is the trunk of the trees and forget that a tree is so much more. Anyways, yes, questions. So if you have a question, I think you can raise your hand or unmute yourself and ask a question, or you can put the question in the chat at the bottom, and I will try to read them out if I see them in time. So are there any questions?
Soren, maybe Carolyn can just give a quick overview of where the button is to raise your hand.
Oh, yeah, that's a good idea. So there is
there's 60 people here.
Yeah, at the very bottom of your screen, if you bring your cursor down, you'll see a whole bunch of little icons, and there's one that's called reactions, and there you can raise your hand, and that's one way to do it. The other way is to just unmute yourself and go ahead and ask a question. I see, Larry, you have unmuted yourself, so I'm assuming you have a question.
Yeah, I've been looking at lichens, but I find them very confusing. I mean, I recognize some. Is there some sort of good identification chart or something for a beginner to identify lichens?
Absolutely. If you can let me share my presentation again.
Yeah, go ahead.
I have a slide where I have some resources recommended. So just jump into that. Hold on, sorry, I have this all scripted out. There we go, books. So there is a free guide to beginners you can download as a PDF document from the provincial government website. The URL is here. I can send that along to Soren as well, so he can send it out via email. Sure. And that has about a dozen, maybe 15 different species that you see pretty commonly, and a couple of rare ones included, just so you don't accidentally grab them. You wouldn't want to be responsible for taking a sample of boreal felt lichen home. There's also a field guide. Francis Anderson, who's a research associate at the Nova Scotia Museum, published with Troy McMullen, a field guide to the common lichens of northeastern North America, through the New England Botanical Garden Press, I think it is. That was going for about $40. If you contact Frances directly, she'll usually still have a few copies on hand she can sell you, but you can also buy it from the New England Botanical Garden store, I think. And I would definitely recommend getting it there over Amazon. I've seen that book listed for $150, $160 on Amazon. Don't pay $160 for it. It is not, it's a great book, but it's not a $160 book. And you can get it from Francis for a lot less, or from directly from the supplier. So there's also a book called The Macrolichens of New England. And that's more of a set of taxonomic keys. But it's a decent one if you're, so that's kind of the next step after a field guide, where you're taking samples home maybe and going through some taxonomic keys and maybe trying a chemical test or two.
Yeah. Thanks. I have that Common Lichens of Northeastern North America, and it's nice.
It's a good one.
It is a very good one. Anybody else? Oh, I see Holly Woodill. Holly, you can unmute yourself and then ask a question there. I just asked you to unmute yourself. There.
No, I don't actually have a question. I just can't figure out how to lower that hand.
Oh, if you click on that thing again, it'll say raise hand and then it'll say lower hand. So if you go back down, then you'll be able to lower your hand. Good. Anybody else with a question? Let's see if there's a question in the chat, maybe.
Holly Woodill, is it? She's had her hand up for a little while.
Oh, sorry, I didn't see that. Yes. No, I think Holly Woodill, she was the one who couldn't lower her hand.
Okay. Fiona, I think, is the next one. Then.
Yeah, Fiona.
Hello.
Here, go ahead.
Hi there. Thanks, Sean. That was super interesting. I really enjoyed it. I was wondering if you were mentioning in your studies a few different things that you were measuring. Was humidity one of them? Are you measuring the relative humidity both from the clear cut inwards? And then I'm also wondering if you're measuring humidity as you move up the tree and if that affects abundance.
Yeah, so we haven't measured relative humidity yet. Katrina, the student on the Canopy Lichens project, would like to measure it at the base and in the crowns of the trees to understand more about gradients and humidity. And I think for the Canopy Lichens project, that's an appropriate thing to consider. But keep in mind, with cyanolichens, we know that they need liquid water. It doesn't matter how humid it is or what the relative humidity is. They need liquid water. It can become photosynthetically active with a dense fog, with rainfall, but 92% relative humidity doesn't cut it. So while we do expect it to be more humid in the forested wetlands than in the clear-cut areas, because a lot of people have shown that when they looked at changes to microclimate after logging before, and we expect a change in the range of humidity over the course of a day in the clear-cut, an increase in the range of humidity over the course of a day. But we haven't measured it directly in the edge influence study yet, partly because we didn't think that it was necessarily going to be, I guess the best way to put it is it's kind of a self-evident pattern, and it's also not necessarily ecologically that relevant when it comes to cyanolichens. If there was a change in the amount of fog created or the amount of through-fall precipitation, the rainfall that goes through the canopy, then that might affect things, but there's obviously going to be more through-fall in a clear cut. So in a way, like it just didn't make a lot of sense to start looking at those microclimatic patterns in a clear cut to interior swamp gradient, because we have there's a lot of intuitive stuff there to interpret. And the other side of it is to get a data logger on each tree would have been thousands of dollars. And so we're always kind of working on shoestring budgets here, and you have to be a little bit selective. I have worked with microclimate data before in some of my previous studies, and basically for every data logger you got to shell out about $80 to $100, and then you have reams and reams and reams of data you have to analyze. So it adds a lot of cost to the project. It adds a lot of time to the project, for which you need additional research assistants or graduate students. And anyways, for a combination of logistical and ecological reasons, I think we just opted to not measure it in the edge influence project. You're certainly, it's good that you asked, though, like thinking about these things being poikilohydric. That's where a lot of people's minds go first off when they hear about these changes. But yeah, there it is.
I have, thanks, Sean. I have two questions that are really quite similar. One by Mary Kennedy, which is what features to include in a photo to facilitate ID? And the other one is how accurate is iNaturalist in identifying lichens when you take photos of them? So I guess if you're using iNaturalist, what's the best photo? How should one take a picture, I guess, of the lichen?
Okay. So I have another slide for that.
Okay, good.
If you don't mind me sharing again.
No, that's good. That's good.
I'll just pop in here. So one thing is I really recommend that you get a hand lens if you're going to be doing any lichen observing. You can actually hold that hand lens right up to your camera and use it as like a little field microscope. Hold it up against the tree within a couple centimeters, and you'll get a magnified image of doing that. A lot of cameras are so good nowadays, you don't need it. But if you have just a basic smartphone or basic camera, you can just hold that hand lens right up to it and get a really good picture. I like to say you need upwards of 5 photos for a proper identification of a lichen. If you kind of follow these rules, there's a good chance you're going to be able to get a very reliable identification on lichens that you post photos of to iNaturalist. If you're just taking the same photo at 2 feet from the tree every single time, half your photos are not going to be identifiable. Either they'll be too blurry or not showing the features that we need to see or what have you. And there are some things, maybe 25% of the lichens you take photos of are never going to be identifiable to a species level, because you need chemotaxonomy to really get that species level identification. But I'd say for the vast majority of macrolichens, we can identify them if we have enough photos. So I like to start with a habitat photo of the lichen, including kind of the position of the lichen on the substrate, maybe showing where it is relative to the tree or the boulder it's growing on. Zoomed in macro photo showing kind of the colony shape of the lichen. You can kind of understand maybe the size of the lobes. It's good to have a scale bar in there too, a little ruler or a pen or a pocket knife or something. This is zoomed in with a hand lens paired up with a smartphone camera. So this is a sort of a microscopic photo of the top of the lichen, showing the details of the ridges, reproductive structures, outgrowths. From this photo, I can tell this lichen has Serratia. That's one of the reproductive structures, little balls of fungal hyphae and cyanobacteria on the margins that makes it look kind of mealy. And with some species, we also need to see the underside. And this is Peltidra caulina. We know from the serratiate margins on the tops of the lobes, from the bottle brush shaped rhizines on the lower side. There's a lot of terminology you kind of get used to as you do more identification of lichens, but basically, if you can kind of follow these features, you're going to get most of your lichens identified. And then if you want to get really fancy, like Frances Anderson, and walk around in the woods with all the spot test chemicals in your pocket, you can do a little spot test and take a photo of that. And we can see from that spot test she did on this Cladonia strepsilis, it reacts to household bleach to turn a green color. So 4 to 5 photos should get a pretty reliable identification on iNaturalist.
Right. Any Buddy with a question before I read another one? Wants to ask a question? Maybe if not, then this is a question about a specific growth on gooseberry. So I have excess thick lichens coating my gooseberry bushes and small fruit trees. They do not appear to be thriving, and I wonder if there are any conditions which leads to this.
Well, I mean, I'm surprised that you have a lot of lichens growing on your gooseberry bushes. They must be very well cared for, gooseberry bushes, if they're getting old enough to have lichens growing on them. um, I think my guess is that what you've got growing on your gooseberry bushes is probably fairly common species, and so I wouldn't be too worried about it not looking healthy. Um, if you wanted to send me a photo of your lichens on your gooseberry bushes at the museum, like one of my jobs is identifying little green things people take pictures of. So feel free to send that along. It's hard for me to offer advice without knowing more about what the lichen is and sort of what the colonization extent is like on the bushes. And yeah, so if you want to send me a photo or maybe a follow-up message to my email at the museum, I can probably help you out a little bit better there. I'll post my email address in the chat for the meeting here.
Great. Thanks, Sean. Excuse me. Any other questions that anybody has? So oh, I see a question. Caroline, you go ahead.
I just have a quick question. First of all, thanks for that presentation. You mentioned pollution as a threat, and I know you said it wasn't like as bad as, say, certain areas in Europe, here in Nova Scotia, but like, is this a good cause for, in terms of, I'm thinking of like how everything is connected and we're always trying to kind of articulate the need for environmental protections and a reduction of emissions, and like, do you see pollution as like an increasing threat for Nova Scotia lichens, and something that we need to kind of do more work to mitigate in the future?
So I'd say in Nova Scotia, the main source of pollution-related problems for lichens would be localized point sources. So if you have a power plant or like a biomass plant, or you have even just a really busy highway, you're likely to get a lot more airborne pollution. And lichens react not just to the sulfur dioxide emissions that are put up into the atmosphere by smelting plants in the northeastern states and Great Lakes regions. They react to nitrous oxides, nitrogen oxides that are released by vehicles on roadways, things like that as well. So in general, close to roads, you see eutrophication of your habitats, which means nitrogen enrichment to the point where it's causing problems. And that's true in lichen communities as well. To some extent, you see these point sources of pollution creating some impoverishment in lichen communities in very, very close proximity to those point sources. In cities, you see lower diversity and particularly lower abundance of very pollution sensitive species. But things are definitely improving. We do have some, I mean, as a scientist, I would always love to see more monitoring. So if we could go from, 10 pollution monitoring stations in Nova Scotia to 40, that would be fantastic. I could create some excellent spatial habitat models, look at the influence of pollution much more effectively if we had those data. But we do see, when we look at the major stations over the last 30, 40 years, that many of them are pretty new stations, but some of the ones that have been around for a long time, there are pretty significant declines in airborne pollution. And tentatively, I would, and it's hard to separate recovery from airborne pollution from perhaps longevity of the habitats or increasing habitat quality in other ways. But we are seeing, I think, in Halifax, for example, more of the typical forest or natural forest lichen communities kind of creeping back into the city. And this is anecdotal. I don't have a large data set showing this, unfortunately. That would be great if I did. But it's consistent with what colleagues elsewhere in the Maritimes have told me in their observations as well, that tree lungwort is a really great example. That's that big green hand-shaped one that you see hanging off of trees in great layers when you're out in the Blue Mountain Birch Cove Lakes Wilderness area or Blomidon Provincial Park, but you get into a city and there's none of it. Well, increasingly on the outskirts of Halifax, we're seeing little patches of it appear here and there. There's one little thallus that I know David Richardson's been keeping his eye on in Point Pleasant Park that seems to be doing okay. and that might not have been there 30 years ago. So the situation is improving a little bit, but one of the things with acid pollution is it has a long legacy of impact. So once that buffer capacity of the soil has been affected, and to the point where trees are being affected by the acid pollution, it takes a long time for that buffer capacity of the soil to recover, if it ever does. We are seeing, people who study this more broadly in Eastern North America are seeing recovery of certain types of ecosystems, particularly those ecosystems that naturally have a high buffer capacity in the soil. They have very calcareous soils, calcium-rich soils. Those ecosystems are recovering the fastest. Ecosystems that are naturally acidic, as in much of Southwest Nova Scotia, for example, those ecosystems have been much slower to recover. So I think things are improving. It's still not, we're not at zero pollution by any stretch. And I think any improvement in monitoring or data quality is always going to make things better for scientists and easier to understand how these things are affecting lichens more broadly.
Thanks. Yeah, I think when it comes to pollution, we are doing better. There's less of it. Years ago, I studied saprophilic beetles related to beetles that live on deadwood in forests, and there are many beetles. It's phenomenal how many there are. But I gave a talk to a German group of foresters who came to Nova Scotia and talking about these saprophilic beetles. And one of the German foresters came up to me afterwards and talked about how in Germany they had started to leave deadwood in their forests, and how many of the beetles that they thought had gone extinct were coming back. So I think nature is phenomenally resilient, that sometimes just because we can't find a particular species, they're probably hiding somewhere. And if the conditions come back, then they will thrive. But I think when it comes to forestry and this eliminating our old growth stands or reducing them to very small pockets, that's going to be difficult for the species that rely on old growth forests to recover, because of, as you mentioned, if there's no continuity in the forest, like the dispersal ability can be hampered. So just because one has a few isolated old growth forests, that doesn't mean that the species there will be able to reach, trees that are getting old. So anyways, yeah, that it's an interesting situation we're in, I think, because our human impacts are beginning to add up. And now with global warming, is there time for us to really discover all these species and know what we need to do to protect them all?
That's why we
Anyways,
need everybody out
and
there
that-
taking pictures of lichens.
Exactly, yes, and of all kinds of other things as well, absolutely, and becoming interested in lichens and beetles and mosses and ferns and whatever, so that there is a greater let's say greater pressure on the powers to be to really do something. Great. I think I don't see any more questions. I see great opportunity for the City Naturalist Challenge, April 29th to May 2nd, and we'll certainly have more information on that. And
I'm Soren, I'd also like to make a plug. I've got
absolutely.
I've got a couple of events happening around the City Nature Challenge where people, if you're interested in lichens, you'd be welcome to come out and learn a little bit and get some practice with them. So on April 30th, I'll be doing a guided lichen and moss hike from the starting at the Prospect Lake Community Center at 9 a.m. That's just about 20 minutes from downtown Halifax. And if there are enough people, so that's going to be through the Halifax Field Naturalists. You'll have to email them to register.
Right.
But if there are enough people, we'll add a second time in the afternoon. And secondly, the Nova Scotia Museum is hosting a microscope cafe on April 7th. That's the second last day for getting your identifications in on the City Nature Challenge. So if you have a sample, a photo, description, hopefully a detailed one, bring it on by, and we'll have a bunch of books and microscopes and people who know how to use the microscopes on hand to help you figure out what it is you've got.
Excellent.
And so the time for that, I think, is noon until 3 at the Nova Scotia Museum of Natural History on Summer Street in Halifax.
Wonderful. We're just beginning to produce our April newsletter. So I wonder, Sean, if you could actually send that information to us.
Absolutely.
And then we'll make
Yep.
sure it's in the news, in the e-newsletter that we send out at the beginning of each month. And yeah, no, that sounds wonderful. Because I think it is good to have a little practice session with somebody who knows what they're doing, rather than just going out and, you know, stumbling around. So thanks very much for a wonderful presentation. This was great, and yeah, maybe we can do something else in the future as well.
I'd be happy to.
That'd
Thanks
be
again
great.
for having me. It's been fun.
Great. Excellent. Thank you so
Thank you
much.
so much.
And thanks
Bye, everyone.
everybody for tuning in and taking part in our March BNS meeting. Thank you. Okay, goodbye, everybody. Let's see, I gotta.